the instruction manual for how to build life. From microbes to plants to human beings, it defines us all. The complete set of instructions encoded in an organism's DNA is called its genome and is passed from parent to offspring during reproduction. Information is stored in DNA using just 4 types of molecule which occur in pairs. There are billions of these pairs, organized in a double helix structure that is both strong and compact. These pairs also allow each strand to act as a backup for the other, a remarkably efficient way to safeguard this precious genetic information. DNA folds into paired packages called chromosomes that are stored in the nucleus of the cell. Different species have different numbers of chromosomes. Humans have 23 pairs. Chromosomes contain many genes. A gene is a section of DNA that holds the instructions for a protein. Proteins are essential for life and perform a huge variety of jobs, from controlling the function of a single cell to determining the shape of a whole organism. Within a species, each organism has very similar DNA. In human beings, the difference between one person and another is a fraction of 1%. But it's what makes us individuals, giving us different facial features, hair color, and height.
DNA. What is DNA? DNA is deoxyribonucleic acid that contains genetic information. It is the ultimate generator for cell and it codes for your trait. And it's basically what makes you, and you.
DNA responsibly controls the cell, and if you make another cell, for example mitosis, you will also have to get more DNA into that.
Let's explain our topic for today, which is DNA replication, which is replicate, making more DNA. However, it takes a lot of steps to understand that.
The purpose in our video is to make you understand in easier and fun way.
Okay, first and foremost, where did DNA replication happen?
It occurs in the nucleus.
But not all has nucleus. So what we are going to study today is DNA replication in the eukaryotic cell.
Oh, but when DNA replication will happen?
It typically occurs during the stage known as interphase. Interphase is a process before the cell division occurs.
DNA replicate does not happen during cell division.
So yeah, before we start, we need to introduce our main character or key players of DNA replication.
One of the major key players is enzyme. Ding. Enzyme has the ability to speed up the reactions. So these other enzymes are involved in DNA replication: helicase, DNA polymerase, primase, and ligase.
Helicase is an unzipping enzyme. If you recall that DNA has 2 strands, you can imagine that helicase will unzipping the 2 strands of DNA like a zip.
DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA.
Primase, the initiator. This is because DNA polymerase cannot figure out where to get started. This is why primase is needed.
Ligase, the gluer. It helps to glue DNA fragments together. DNA replication starts at a certain part called the origin.
This part is identified by certain DNA sequence. There can be multiple origins within the DNA strand. Let's get started.
This is helicase. Okay, so helicase will come SSB protein is single-strand binding protein. The function of SSB protein is to keep them separated. This is primase. Primase comes in and make RNA primer on both strands. This is really important because others DNA polymerase would not know when to start.
DNA polymerase. So now you have two strands of DNA, right? They are not identical. Remember, they are complement to each other and also anti-parallel, so they don't go in the same direction. Later, we are going to explain regarding 3 to 5 prime and 5 to 3 prime direction in this video.
DNA polymerase can only work in 5 to 3 directions, so the strand that runs in 5 to 3 direction is called leading strand.
But the other strand will make it a little tricky. DNA polymerase can only go in the 5 to 3 direction.
So, primase has to set a lot of extra primase down to do that. It takes longer time too. This strand called lagging strand.
On the lagging strand, you tend to get little fragments of synthesized DNA. They are called Okazaki fragments.
This is like this. The glues enzyme has to take care of the gap in the Okazaki fragment.
Therefore, at the end of the process, you will have 2 daughter DNA molecules from your one parent's DNA molecule. It's called semi-conservative. But why is it called semi-conservative?
Because there are 2 copies of each contain one of the original strand and one from the newly made strand. Before we end our video, let's get back to the explanation regarding DNA strand direction.
The direction for the DNA strand are different.
DNA either goes 5 to 3 or 3 to 5 strand.
But what does it mean?
Well, the sugar of DNA is part of the backbone of DNA. It has carbon.
The carbon on the sugar are numbered right after the oxygens in the clockwise direction. 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime.
The 5 prime carbon is actually outside of the ring. Now, you do the same for the other side to keep in mind that the strand is flipped. Just because DNA strands are anti-parallel to each other